Hi everyone, this is Jason, producer of Data and Biotech. Before we get started, I wanted to let you know about our latest white paper. It's a comprehensive guide to implementing machine learning models in biotech manufacturing. It's a complete overview of all the potential problems of ML adoption and, more importantly, how to solve them. To download it, simply visit connect.cordine.com/biotech-ML. We've also dropped the link in the show notes of this episode. Okay, let's get into it. Welcome to Data in Biotech, a podcast from Cordine where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. This week, we sat down with Wolfgang Halter, head of data science and bioinformatics at Merck Life Science, a leading global science and technology company. During the interview, we discussed the BayBE project at Merck, an open source library they have built for Bayesian optimization, and how it's used for experimental design and to accelerate innovation across the group, in some cases offering time savings of 50%. The pair discussed the benefits and challenges of Bayesian optimization, including the need for standardized data models, and Wolfgang also shares some advice for young scientists and engineers in the biotech industry to get ahead. Here we go.
Wolfgang Halter, welcome to the Data and Biotech podcast.
Hi, Ross. Thanks for having me.
Awesome. Well, just to kick us off, could you just give us a brief overview of your background and career to date?
Sure, absolutely. So I studied engineering cybernetics at the University of Stuttgart. I focused during my studies on systems biology, did my PhD in synthetic biology, also in Stuttgart with a few stays abroad. And since 2019, I started as a data scientist at Merck for the life science business of Merck. And today I'm leading the data science team and the bioinformatics teams since 2021.
Awesome. So why do you do what you do? What motivated you to take this path?
It's a good question. I think it's like an urge for efficiency. I know this is like very stereotypical, I guess, but I really have the feeling that like there's so many like valuable insights, often like times covered in data. And for me, it's a real pleasure to unpeel those insights and to like leverage them to make things like just work more smoothly and more optimal in some sense.
Awesome. And so can you just talk a little bit about the mission of Merck Life Sciences as well? What kind of work does it do? And then also, how does it relate to the Merck group more broadly? Might be helpful as well.
Sure. Yeah. So let's start with the Merck group. So the Merck group is divided into basically like three sectors. So we have like an electronics business, a healthcare business, and the life science business. And for Merck Life Science, the main mission is really to impact life and health with science, basically. And that means like we are a service provider as well as like producing products for like the life science industry, for other pharmaceutical companies. And we envision a world where like our innovative products and services and digital offerings as well help to create like solutions for people. And we want to do that like across the world and to basically ensure like a sustainable future for generations to come. I think that's like the main vision of the company. That means also like we work usually like in a very much like partnership focused relationship with our customers, with our patients. And with different goals in mind, like working on life-saving therapies, diagnostics, and preventative solutions, as well as like ensuring like scientific breakthroughs and tackling like the really hard challenges of research and development. It's all about like challenging the status quo and to be innovative in everything we do.
And so, as a data science team, how do you support that work?
So the data science team here at Merck Life Science, we are embedded into something that's called like Analytics Center of Excellence. So we are like really a hub around data and analytics in general. So we have like lots of teams that are also supporting the whole journey around data analytics, from data ingestion to data governance to creating insights using that data to creating applications that then help like solving certain problems in the business. And the data science team itself, we really support that journey and with a focus on like the creating insights on with the data that we gathered. In particular, we are trying to craft AI and bioinformatics solutions to accelerate like innovation in life science. I think that's like the core of our part. And then like one very important side business, so to say, is like also providing skills as a service to the whole company whenever they need something like that, and to upskill the people in the company to foster like the data centered view in everything we do.
Interesting. So so as a center of excellence, you're charged with disseminating insights directly, but also helping to build tools and embed capabilities in different parts of Merck Life Sciences that enable the business to drive insights and improve efficiency more broadly. Am I thinking about that right?
Yeah, absolutely. Yeah.
Awesome. So, I know that your team has been working recently on a tool related to design of experiments, DOE. Mm-hmm. So, previously we had Marcus Gershader from Synthase on to talk about DOE. And, so I'm interested in the work that your team has done related to DOE. And just, I know that you've been working on an open source tool called BayBE. Am I pronouncing that correctly?
Yeah, we call it our baby. So yeah.
Okay, yes. So if you wouldn't mind just talking a little bit about how your team integrates with design of experiments more broadly at Merck Life Sciences and how that led you to the development of BayBE.
Yeah, that's a good question. So BayBE stands for Bayesian backend. So, and that also already tells like what it is about. It's really about like at the core, it's a toolbox around Bayesian optimization, and bringing that together with design of experiments is already showing you a little bit like what our way of thinking is in that area. I think we started our first design of experiments use cases like 4 years ago when I also started the company, and there were probably like more of them around before I was there even. And like my colleagues and I, we were coming like more from like the scientific and academia world. So we were also thinking like, why are people still doing like this old-fashioned design of experiment things? Like there are so much better methods or better approaches available these days. But when you look into like the offerings, public offerings like vendors and softwares, professional softwares for that, you don't find too much going beyond classical DOE. And that's like where we started developing like more probabilistic based approaches, more iterative approaches where you take into account like all the information that you have about your system that you're trying to optimize with each optimization step. And I think that's at the core of BayBE, so to say.
Interesting. I think it's worth spending a little bit of time on, what were the classical approaches to DOE and why is the Bayesian approach, the right alternative to that?
Yeah. So for me, the classical approaches are the ones where you look at like maximizing your information content, and you basically design your experimental space just based on the parameter space that you have. But you usually don't take into account like what kind of experiments you already did in that area. So, and if you do, like you always do like then a full factorial or something like that. Either way, like with just like a limited or like a new parameter space that you're looking at. Now with BayBE, it's different because we really kind of incorporate like all the knowledge into the priors that we have about the parameter space. And that really gives us like a predictive model about like where we expect the highest outcome at what probability. The key here is really including probability measures in your design, because that allows you to like do like a really nice balance between exploitation and exploration, where you say, okay, I really expect something to be like much better than that I have experienced so far in that area, but another area also looks promising, so I'm going to focus on those two, while like those where like it's just like very unlikely you don't do any experience at all. So it's really like this incorporation of uncertainty and previous knowledge about your system. That's the difference here.
Interesting. So I'm going to try to say back to you what I heard to make sure that I'm understanding correctly. So in classical DOE, you had a sense of what your parameter space was. So in the parameter space in this case is like, what are the variables that you can change in order to influence the experimental outcome that you care about. So, I noticed in BayBE, you're targeting yield from some, for something along those lines. So, the classical approach is, sort of is picking points inside of all of these different parameters that are sort of widely spread so that you can see, broadly speaking, in a static view, what does the space look like? And then choose kind of the optimal point in that space and work from there. Whereas on the Bayesian side, what I hear is that there's this iterative approach that you can take where, set up the parameter space, but then you're sort of gaining information about different relationships between these parameters and your targets, and also be able to quantify your uncertainty about where in that space might be worth exploring. And then you can sort of select the next best experiment or set of experiments that based on, where you want to resolve uncertainty. So where you want to explore, exactly. And then where you want to, exploit what you know already, where you want to continue to optimize toward the best possible set of parameters. Am I thinking about that right? That's
exactly right. Yeah, absolutely right. And I think like when you look at it in terms of like batches, usually like in a classical way, you do like a whole design of an experiment, then you have to test like your entire design, like depending on how well you want to cover your parameter space. That's a lot of experiments usually. With Bayesian optimization on BayBE, the best approach actually, if you can afford it and if your experiments don't take too long, is to really go iteratively, like step by step. Do one experiment, then look at the next one, and then do it like really step by step, and let the algorithm guide you through that parameter space. And then that really ensures like the best balance between this exploitation and exploration phases. And it doesn't mean that you can't do like batch experiments. Like oftentimes we work with like 96 well plates, and then, you have the space. Why shouldn't you do it with like 96 experiments at the same time? And it's also good. It helps speed up things. But like this Bayesian approach is then allowing you to like do really from batch to batch a huge information gain.
Interesting. So in this context, we're going iteratively, one experiment by experiment would somehow be optimal in terms of resource utilization, but there's also a speed component here too, where you might want to parallelize a lot of these experiments. Or, you mentioned like a 96 well plate, you've got machines that only do 96 experiments at the same time. And so you might as well sort of leverage that capacity in order to optimize your throughput. Are there other considerations that you like bring to the table when you think about, I'm imagining advising different units within Merck Life Sciences about how to approach Bayesian optimization and, when to parallelize versus when to be more sequential in the experiments?
Like it really depends on like how long it takes for one experiment to look at, right? I mean, if one experiment takes a few minutes, then it's not necessary to do like ultra parallel. However, if you are looking at like, stability experiments where you put something in a freezer for 3 months and then test it afterwards, that's your limiting factor in the end. And then you have to treat those differently. You can't do that sequentially in the end, right? So that's like, I think that the main deciding factor for how you set up then your sequential versus parallel design. And in the end, like the upsides are really mostly about speed, getting to better results faster with less iterations. So that's like the main point about this. And as a side note, it also helps to save a lot of resources. As you said, like ideally you spend also like less chemicals and less materials in your experiments. And that's also something that would define like your approach. How much importance do you want to put on resource saving versus time saving? So you have to choose your weights in a multi-objective optimization problem, basically.
Interesting. So, in order to sort of clarify things before I ask more about sort of the benefits and, how it's being used internally, could we take an example experiment or, I know that you use sort of the construct of a campaign, an example experimental campaign that might be done at Merck Life Sciences or some other place, and sort of how a person or a team goes about thinking through how to structure an experimental campaign in BayBE, and where the insights come from through that process.
The general points are always the same. Like in the end, it starts with like optimization problem, right? So you want to make something more optimal. Sometimes that could be like the formulation of a therapy, or it could also be just like even in a digital space, like fitting the parameters of a digital twin model or something like that. So it could be either. So that's like your starting point, right? And then the first step is really figuring out the search space. So what are your parameters? What are your variables that you can vary to influence your target? Let's take cell culture media, for example, that's like a very nice example. You have like just very many components in cell culture media that you can vary. So it's like many parameters at the same time. And then most of the time, even like on a continuous scale. So it's not even like choosing 0 or 1, but like choosing any number between 0 and 1, basically. So the search space is quite big and a mix of continuous and discrete variables. So that's like usually not a very easy problem to solve. Then if you have your search space, you're looking at your objective again. Oftentimes, like we start with, we want to create the best cell culture media. At some point, you need to formalize that and think about like, okay, what does it mean? Like what means best in this place, right? Like, and really defining those objectives is actually not as easy because like you have like oftentimes like many different objectives that are competing with each other. So in many cases, you end up with like a multi-objective optimization where you have to really then define, okay, like what's the most important objective for you and how do we want to design this in the end?
On that objective front, is that thinking about like that as quality and quantity, for example? So, making more of something might mean you make a lower quality of something. And so you sort of need to weight the two objectives against each other and, help the model understand how to balance quality versus quantity in the objective. Am I thinking about that? Right?
Yeah, it could be. So for example, I mean, cost versus like quality, it could be like those two objectives like competing to each other. Like, for example, like if you choose like raw materials that are like of lesser quality, but they are like cheaper, and you may achieve like the same outcome in the end at a lower price. But for us, I think like we didn't encounter that too often, to be honest. I think like most of the time we're looking at things like ensuring pH in a certain range, but at the same time you want to have like a certain activity of your proteins, and like pH and activity also like influence each other. So that's like the different objectives, and it could be like a quite big list actually in the end that we're facing. We have the search space, we have the objectives, and then basically we translate this using BayBE. It's a super simple interface. You translate this into like an optimization problem, and then you just get started. Basically, you get your first recommendation for your first experiment. People conduct that first experiment or like first set of experiments. They play back the results into BayBE, and they get the next round of recommendations.
Is there an endpoint? How do you know when you're done with this kind of process? Or is it just that, you reach some threshold where you feel like this is sort of good enough?
That's a very good question, because that's also like one of the advantages of Bayesian optimization. You can actually get this measure of like how much information gain can you expect with additional experiments. That's also something that you can't get from classical DOE. Like in classical DOE, you can say, okay, maybe I just need to make my search grid finer and I conduct even more experiments, or I need to extend my search space somewhere. And with Bayesian optimization, you usually get like a good measure for how well you covered your search space already and how likely is it that you will find something that is even better. So that's actually a feature that we're working on right now to also implement these dynamic stopping criteria, so to say.
Interesting. I'm assuming that you developed this open source library because you were already doing these sorts of things internally. You thought that, having an ecosystem around this would be valuable. Could you share a little bit about why you decided to open source this?
Yeah, it was a process, certainly. I mean, we started, as I said, with one design of experience use case. We made it a Bayesian approach, or we tackled it with a Bayesian approach. After that, we encountered like one or two more of those kind of use cases. And then we reached out in the company, we realized, okay, that there's a huge potential for these kind of applications and use cases, but each of them need to be like tailored in one way or the other. So we initially, it started out of just like getting more efficient internally to be able to scale and deliver use cases in a faster fashion. And then at some point we said, okay, this is not just for us. We also can partner up with like academic institutions, and we can also get some input from other partners that are working on these kind of problems. And for us, that was basically the idea then to go open source and to share this with the community, to also get input from the community.
And has that input been valuable? Has it, been formative in the tool to date? Or are you mostly hoping that, over the course of time, it will continue to help the tool develop?
Yeah, I mean, we open sourced it in December of 2023. So it's really relatively fresh, I would say. But we have been working together with the Acceleration Consortium in the past already, even before open sourcing it. And we already got like some really valuable input through them. And we are hoping that we can expand that even further.
So turning back to BayBE and how it's used internally, what has the adoption been like from data scientists on your team, from biologists or bench scientists across Merck Life Sciences? How's it being used today?
There are different levels of answers to that. I think like when we look at at core, BayBE is a software development kit. So it's really made for data scientists to develop things. And that means like we have like this of developers of applications that is still there. But every application that we roll out using BayBE is adopted like very fast and really well. And we have like a long list of projects that are waiting to be implemented, basically. Even though, I mean, BayBE is speeding things up so tremendously. It's like we collected quite a few projects in terms of like demand. What we also see is that like since we open source, we also allowed like OEMs to work with it for things that they develop with us, for example. And there we also get like really good feedback in terms of like how easy it is to use and how easy it is to implement. And that has been like very valuable for us and showed us that we are on the right track.
Are you able to share any examples of kind of the applications that you're developing? I'm interested in just like, obviously you call it Bayesian backend for a reason. So imagining there are a variety of different, it's a toolkit for developing the backend of an application, but then there's a variety of different front ends or use cases that that people might see. So any examples would be really interesting.
I would say one example is a product that is based on finding viscosity reducing excipients. So it's excipients that you put into your formulation that reduce the viscosity. And you need that to just like being able to administer drugs in the right way. And we basically developed a tool that allows you to test just a few excipients with your protein. And then it suggests you ideal combinations of those excipients to find like the best excipient combination for your protein that you have and that you're trying out. And this is something that we offer our customers to use, because we are also like distributing like some of those excipients. And if you would have to test like all combinations of those excipients, it will take a lot of time and effort and a lot of resources. So we try to minimize that so that to allow our customers to speed up and go faster to market, basically.
Interesting. So, it's a way in which you're enabling your customers so that they can scale faster, and that has benefits for you as well. So it sounds like time savings, both internally and externally, are sort of a big value. Do you have any estimates of like how much time you're able to save in a given domain, or is it just difficult to estimate?
We have certainly like some experiences, and also you can actually find numbers in the literature. Some numbers in literature claim that like this approach would lead to like 95% time saving. What we experience is more like in the area of like 50%, which is even then huge if you think about it. And I would say like the minimum is really like 30%. So 30 to 50% is something that we consistently see in the projects that we conduct.
Interesting. And, as you have different customers and business partners coming to you and asking for applications of Bayesian optimization to their particular problem space, how do you think about prioritizing which of these use cases you're going to go after? Or, is there a way to sort of like bundle multiple use cases together in a way that's smart for your team?
Yeah, I mean, like the latter is always like what we think about first. How can maybe we can serve someone's need with an application that we already have developed for another group. And oftentimes this is already the case. And that's like the best case scenario. The other needs is something where we really look at like what's the potential impact of that solution. Like really we try to prioritize based on how we can create the most value for our customers and the company in the end.
Interesting. And I'm imagining that, having all of these different campaigns and the data that these campaigns collect is really valuable intellectual property that you're developing. And I'm wondering, do you somehow store for later or leverage or combine or analyze the data that these experiments are throwing off in a way that yields more insight? And if so, how do you approach that?
So in principle, like any experiments that are conducted in the lab are gold in that sense, right? It gives you some snapshot about like a little bit of truth of the world, and you really want to conserve that in a good way. And I think that's like one of the biggest pain points in large corporations and like with many different labs to store that data in a meaningful and also intercompatible way. so that you can make sense of it like as a whole. I would say we're not there yet. That would be my vision, that you really have like this uniform data model in terms of like research and scientific insights, where you can just like feed all your experimental data in, and in the end you can just throw BayBE on it and you will get like the perfect recommendation for any problem you're looking for. That would be great. We're not close to that vision at all. But what we see is like for the applications that we have developed, like design of experiments, and we see like these individual campaigns where BayBE is running. Like one of the key features that we're now just rolled out is like really transfer learning. That means like learning from past campaigns. Maybe like when you do this viscosity experiment, when you look at like proteins that are similar to that, you can learn a lot like about like how proteins behave with those excipients. And you can basically get like a warm start for your DOE. And maybe that's not the best example, because proteins really behave very crazy sometimes. But in other domains, when it's like cell culture media, for example, or also like for model fitting, like these warm start features are creating, like that's where we get from the 50% to the 95% time saving.
Right. And so are the conditions for that kind of transfer learning that parameter space and the objective have to be the same, or at least similar enough? Or, how do you think about when transfer learning can be applied to a given campaign?
Mm-hmm. Yeah, for us, I think like right now, the status quo is that the parameter space, at least in terms of like what kind of parameters you have, maybe UX space, but at least the physical properties are the same. That's basically the condition right now, I would say. You can extend it to then unseen parameter spaces. That's also possible, but you need to have like some overlap. Otherwise, like without the overlap, then you don't gain anything. That's certainly like the main limitation right now. But I think that's not something that you can fix.
Yeah, that makes sense. And is transfer learning in this case being used in the same case that we use it with regard to neural networks, where you're literally taking sort of an optimization algorithm that's a neural network, and then transfer learning that optimization algorithm to a different campaign? Or is this transfer learning in another context?
It's basically, you can think of it as like you're changing your priors, like your prior information about your parameter space. Like when you don't know anything, you have to start with usually like some uniform priors, and you have to work a little until you're like you just like get some more information about your distribution of parameters and in the best way. And then if you start with like non-uniform priors, then you can already look in the right regions.
Right. So just to summarize what you said and make sure that I'm understanding, it's that in Bayesian optimization, you start with this idea of a prior, and a uniform prior is this idea of we know nothing right now. The answer
Exactly.
could be anything within the parameters, anything anywhere within the parameter space. But if you already have a set of priors about where in that parameter space the optimization is likely to be, then you're going to get there a lot faster if you start at a place that is closer to where the optimum is. But because Bayesian optimization is so powerful, even if your prior is completely wrong, even if the transfer learned prior is wrong, all that's going to lead to is just the need to gather more data. And eventually Bayesian optimization will get you to where you need to go.
Will adjust.
Yes, but transfer learning can get you a lot closer, a lot faster, which to your point leads to more time savings. Am I thinking about that right?
Yeah, absolutely. I mean, you can think about it like in terms of when you try to optimize your way to work in the morning, and you think about like what routes work, how fast, and like it really depends on traffic in the end, right? But like your experiments, if you know nothing about like that city that you live in, either way it won't matter because you don't have any experience. Like they might be the same length, but you don't know if one way is more clogged than the other. If you live there for some time, you get a feeling for it that like on Fridays you should probably take like the highway on Wednesdays you may want to take like the small roads. And that kind of like knowledge is exactly what those priors are. They give you like a first idea about like how it usually looks like. It may change. You may get on the road and then you see at the first crossroad, hey, this is not going to work today. There's like a parade somewhere. I need to divert from that. And that's exactly what Bayesian optimization will then do. Like after your first point that is basically outside of like what your prior suggests, it will pivot.
Right. So there's exploration and exploitation happening. So even if you're in the wrong place, the exploration will lead you eventually to the right route, to use your metaphor. This method of optimizing or, of learning from experiments just seems like the clear best path of the methods that we have available today, which makes me wonder what has stood in the way of adoption of Bayesian optimization in this context previously? Is it just that the open source toolkit hasn't existed before and now it does? Or is it something else that I'm missing?
I think there are several factors to that. And I would have to say even classical DOE is not utilized to its full potential when you look at like how people in the labs do scientific experiments. So I think like user interface and the gap between the developers of DOE methodologies and the ones who are using it in the end, that's probably like the biggest challenge for DOE in general. And I believe like tools like Synthase that you had like earlier on this podcast can help significantly to lower that entry hurdle, so to say. I think that was certainly like one of the biggest bottlenecks in the past. For Bayesian optimization in particular, it's also something that comes with the computational power we have available these days. So when we're looking particularly at transfer learning, you need computational power. You need to cover like when you have like many parameter dimensions and you take into account like all the data points that you have collected in the past, like the standard Gaussian process approach is just eating up memory and computational resources. That's also something like that we are facing still, like even now with like the seemingly unlimited cloud resources that you have, we see, okay, it is computationally hungry. And we're working on solutions for that to make it more efficient, particularly for like larger domains, like with many data points and still being able to do transfer learning. But yeah, I think that is certainly like also one reason why it didn't get like that much attention maybe 10 years ago. But then again, I mean, there's so many things where you in hindsight think, yeah, why didn't they do it from the beginning, right?
Right. Well, yes. And I mean, also, I know that there's cultural aversion between practitioners of frequent statistics and Bayesian statistics. And so there's probably an element of that in there as well. And, sort of like the extent to which classical methods have a handle on people's sort of viewpoint of the world. So with the time that we have left, I'm just interested in zooming out a little bit and getting your perspective on, what you think are the biggest challenges that are currently facing the biotech industry at large regarding data handling, data analysis, data science in organizations like yours.
I think we touched on that earlier a little bit in terms of like this universal data model. That's for me, the vision that I would like to get to. Right now, we are seeing still way too much Excel, like Microsoft Excel. So like there's a lot of manual data collection being done still in the lab. And I think like this is really hindering like some of the advances, particularly when you look at like cross-lab insight generation. That's something that I would see as one of the biggest challenges, like in general, looking at digitalization in the lab. Like the electronic lab notebook, for example, is something that many people treat as like an electronic form of paper. But it can be so much more. It's not just like a different way of like writing things down. It needs to be what I was telling earlier, like it needs to be this connecting layer between your information. And for that, you need to treat it also like in a different way than just like jotting down information. You need to bring a little bit more structure and standardization into those processes, I believe. So that's certainly like one problem. And there are data standards for like experimental data, but I don't see that like we have reached a good level of adopting those data standards. There are maybe also like a few competing ones. All the vendors that are out there creating like lab equipment, they like each is basically following a different standard or different interfaces and APIs. That's just complicating this vision of like a connected data layer for the R&D world.
Yeah. So, I mean, I've heard a lot about the idea that automation in many respects can sort of resolve this problem of a consistent data model, because at least, the machine that you have is collecting the data points that you need, but that's sort of predicated on the idea that all of these different machines that you use for automation will come together on a data model that's at least relatable or combinable together.
Exactly.
Yeah, and I'm imagining when you talk about ELNs, you're talking about sort of more structured metadata about the experiments that are happening versus sort of the unstructured, like writing it down on paper sort of thing. Is that
exactly.
right?
Yeah, absolutely. And I mean, I agree. Automation can be like one of the solutions, but I think this is a chicken egg problem somehow, because like also for automation, you need to have like a certain level of standard and the right interfaces that can talk to each other.
Interesting. And I know you come from an engineering background, and you come into the field of biotech R&D. Are there how do you think that sort of colors your perspective on the problems that biotech R&D faces here?
Yeah, that has been very surprising to me, to be honest, at the beginning. Even during my PhD, I pivoted a little bit away from just like regular engineering into like also like this biological sciences. And for me, it was very different because in these natural sciences in general, researchers really think end to end. They think of like their research problem from the very beginning, from the atoms to the therapeutics sometimes. Like it's really very well integrated thinking that the natural scientists have and conduct. And I think this is really good because like you really go deep on your topic. What it prohibits somehow is that you create interfaces. And that's something that I'm like coming from engineering, like the key dogmas of engineering. And I think why Industrial Revolution worked out so well is like these concept of modularity and orthogonal design, like designing things in complete isolation of like the neighboring modules, so to say. And being able to do that allows you to just like narrow down on just your module, looking only at that, and then becoming excellent in exactly this module, and then just feeding everything you have like outside to like your neighboring module. And this kind of like modular thinking is something that I believe is missing in the natural sciences a little bit because of this end-to-end thinking. And that's something that I wish we can also move into more in the future. And that's also like probably coming back to like the data standard. It's exactly the same thing. Like you need to have like standardized interfaces, and then you can stay within your domain, do whatever you want, as long as the interfaces don't change.
There's so much there, and I feel like I could have an entire other podcast about just this topic alone. But those insights are really interesting to me. I mean, it's almost back to Adam Smith and like this level of specialization and how that enables the creation of value at different parts of the value chain. Just as we wrap up, do you have any advice for young scientists and engineers who are looking to learn more about biotech and contribute to its growth?
That's a good question. I think we still have too many siloed disciplines and on one end. So even if you are studying biology or chemistry or even data science, I would encourage people to go out and look at like how other disciplines are approaching things. Like it's still astonishing to me how similar certain fields of research are with just like a different language. So that's certainly something that I would encourage people to do, like get some inspiration from other industries, from other areas, look at like how they solve problems, look at what their biggest problems are, and try to understand their language. I think that is like really important. For example, as a natural scientist, I would particularly look at like how does engineering approach problems, how does software engineering approach certain things, like what I mentioned about like this modularity. This is nothing new for like a software developer, where companies like Amazon are preaching the API first approach. This is nothing else but exactly like the modularization of your designs. And I think we can learn a lot from that in the natural sciences as well. Yeah, maybe coming back to the ELN thing, don't think about digitalization as a process of replacing paper. It's much more powerful than that.
Fantastic. Where can people learn more about you and your work and Merck Life Sciences?
Yeah, I encourage you to check out like our homepage, merckgroup.com. We are also like present on like several like social platforms like LinkedIn, or particularly look at our GitHub account, which is EMD Group. There you can find BayBE. You can check it out, clone it, adjust it. You can find also like a few other libraries there. Feel free to give that a look.
Great. Yeah, well, it's BayBE, BayBE, Bayesian backend, and we'll put the link in the show notes and in the blog we write about it as well. Wolfgang, thank you so much for joining us today. It was a really insightful conversation, and look forward to connecting down the line.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.